This hearing will come to order. Without objection, the chair is authorized to declare recess at any time. Before I deliver my opening remarks, I want to note that the committee is meeting today virtually. I want to announce a couple of reminders to the members about the conduct of this hearing. First, members should keep their video feed on as long as they are present in the hearing. Members are responsible for their own microphones. Please keep your microphones muted unless you are speaking. Finally, if members have documents they wish to submit for the record, please email them to the committee clerk, whose email address was circulated prior to the hearing. Good afternoon, and welcome to today's hearing on biological research at the Department of Energy, where we will hear about how these capabilities are being leveraged to respond to the COVID-19 pandemic. I want to thank Ranking Member Lucas, members of the Energy Subcommittee, and our witnesses for joining us today. Members of this subcommittee are enthusiastic about the energy innovations that are coming out of DOE's national laboratories, and rightfully so, given that the labs have provided our country with breakthroughs like supercomputing, inventing new materials, pioneering efficient power lines, improving automotive steel, and discovering 22 elements. Yes, the periodic table would be much smaller without the national labs. As the COVID-19 pandemic began to unfold in the U.S., it became apparent that DOE's laboratories and programs were also well positioned to help us respond to the virus. It is perhaps not well known, but this territory of research is not new to the labs. In fact, as an example, national lab scientists developed a non-toxic foam that neutralizes chemical and biological agents. It was this foam that was used to clean up the congressional office buildings and mail rooms exposed to anthrax in 2001. Lab scientists are also credited for developing the field of nuclear medicine, producing radioisotopes to diagnose and treat disease, designing imaging technology to detect cancer, and developing software to target tumors while sparing healthy tissue. DOE labs house and operate national user facilities like the Joint Genome Institute, established by the department in 1997 as part of the Human Genome Project. Today, Institute researchers survey the biosphere and characterize organisms relevant to the DOE science missions of bioenergy, global carbon cycling, and biogeochemistry. They also provide advanced sequencing and computational analysis of genes related to clean energy generation and environmental characterization and cleanup. Leveraging these capabilities has enabled researchers to develop countermeasures against the novel coronavirus, like diagnostic tests, and allowed them to assess transmission and evolution dynamics as the virus spreads globally. This hearing will examine the historic reasons for why the department possesses advanced bioscience capabilities to address the nation's great challenges and to stimulate innovation, how this expertise and DOE's biological research tools are being leveraged to respond to the COVID-19 pandemic, and what future directions for the department's biological system research can provide solutions for our nation's most pressing issues. I look forward to hearing from our witnesses, sharing their expertise on these topics, as well as hearing how the Science Committee can best support DOE's biological research activities to unleash the next generation of innovation. Before I recognize Ranking Member Lucas, I would like to take a moment to acknowledge that we are holding this hearing on the 19th anniversary of the September 11th attacks, and to ask for a moment of silence for us to remember and honor those who lost their lives, those whose lives were forever altered, and our first responders, the brave men and women who rushed in on this day to help our fellow Americans. Thank you. I'll now recognize Mr. Lucas for an opening statement.
And as soon as he gets control of his mouse, he'll speak. The challenges of technology. Thank you, Chairwoman Fletcher, for hosting this hearing, and thank you for all our witnesses for being with us this afternoon. During all the challenges and the uncertainties of this pandemic, one thing has stood out. Our scientific community has gone above and beyond in the effort to understand, treat, and prevent COVID-19. The Department of Energy and its Office of Science and National Labs have been central to this effort. Today, we have the chance to narrow our focus to DOE's biological research efforts, in particular, the Biological and Environmental Research Program, BER. BER is a high priority research area within the Office of Science that has consistently received bipartisan support from this committee. From examining the complex behavior of plants and microbes to developing new approaches to characterizing genomic information, the BER portfolio helps address today's public health challenges while preparing us for the next generation of bioscience R&D. Much of this work is carried out through BER's user facilities, including the Joint Genomic Institute, the preemptive facility for sequencing plants and microbes. Originally created to lead DOE's role in the Human Genomic Project, JGI sequencing and analysis analyzes more than 200,000 billion bases of DNA each year. 200,000 billion. That's a huge number. Another key BER user facility, the Environmental Molecular Sciences Laboratory, or EMCell, offers over 50 premier instruments and modeling resources to assist researchers in understanding complex biological interactions. EMCell also offers access to high-performance computing resources to support advanced experimental research in these biosciences. Dr. Kelly Wrighton is here with us today, and her work makes great use of the BER resources. Dr. Wrighton is an associate professor of Colorado State University and a recipient of the Presidential Early Career Award for Scientists and Engineers. I look forward to hearing more from her on the value of user access to BER's resources. BER user facilities, along with the other 25 user facilities maintained and operated by the Office of Science, are vital tools of scientific discovery and important drivers of national economic competitiveness. No other system in the world grants this kind of cutting-edge technology access to tens of thousands of researchers each year. Other countries have taken notice. Developing the most advanced scientific facilities has become an intense international competition. The nation with the fastest supercomputer, our most complete genomic data set, or, for example, will hold a distinct advantage in nearly every field from material science to predictive atmospheric modeling. Office of Science programs like BER need robust federal support for large-scale user facilities, which academia and industry simply cannot afford. This is why the key component of my bill, H.R. 5685, the Securing American Leadership in Science and Technology Act, is a competitive authorization, comprehensive authorization of the BER program, which includes a user facility development program and authorization of important initiatives like the bioenergy research centers. This legislation also doubles funding for the entire Office of Science over the next 10 years. This significant investment is essential to U.S. leadership in biological and environmental research. Whether it's COVID-19 or the next public health challenge, our understanding of these complex systems is dependent on the basic research conducted by BER and the Office of Science. I urge my colleagues on both sides of the aisle to join me in focusing our limited legislative days on these bipartisan programs. I once again want to thank our witnesses for being here today, and I look forward to a productive discussion. And thank you, Chairwoman Fletcher, and I yield back the balance of my time.
Thank you very much, Mr. Lucas. I will now recognize the chairwoman of the full committee, Ms. Johnson, for an opening statement.
Thank you very much, Ms. Fletcher and Mr. Lucas, for holding this hearing today, and thank you all to the witnesses for being with us. Today, we meet to discuss the groundbreaking bioscience research supported by the Department of Energy's Biological and Environmental Research Program, and how these capabilities are now being used to better understand the novel COVID-19 virus. DOE stewards many unique facilities related to the biosciences. They range from the department's world-class genomic sequencing tools that have been decades in the making, to large X-ray light sources that can be used to identify various characteristics of and treatments to the virus. Combining this experimental knowledge with the department's state-of-the-art supercomputing capabilities provides our nation with a scientific test bed that is second to none. This extensive biological research portfolio has been leveraged as part of a broad department-wide initiative called the National Virtual Biotechnology Laboratory that was created to help address the issues we face from the current global health crisis, as well as those that we can expect in the future. Not only are the activities of the Biological and Environmental Research Program so critical for better preparing us to respond to potential future pandemics, but also for our national energy security and for addressing the climate crisis. Among other applications, research carried out under this program will help us develop the low emissions biofuels of the future, which will be very important if we work to decarbonize the transportation sector and other parts of our economy. Today, however, our focus is on the program's contribution to the fight against COVID, and I look forward to our witnesses' testimony. I thank you again to our witnesses for being here, and with that, I yield back the balance of my time.
Thank you, Chairwoman Johnson. If there are members who wish to submit additional opening statements, your statements will be added to the record at this point. And at this time, I would like to introduce our witnesses. Dr. Mary Maxon is the Associate Laboratory Director of Biosciences at Lawrence Berkeley National Laboratory, where she oversees Berkeley Lab's biological systems and engineering, environmental genomics and system biology, molecular biophysics and integrated bioimaging divisions, and the DOE Joint Genome Institute. Prior to joining Berkeley Lab, Dr. Maxon worked in the biotechnology and pharmaceutical industries, as well as the public sector in such positions as Assistant Director for Biological Research at the White House Office of Science and Technology Policy, where she developed the National Bioeconomy Blueprint. Dr. Deborah Mohnen is Professor of Biochemistry and Molecular Biology at the Complex Carbohydrate Research Center at the University of Georgia. She has studied plant cell wall synthesis, structure, and function for more than 30 years and currently serves as research domain lead for integrative analysis and understanding within the Department of Energy-funded Center for Bioenergy Innovation. Dr. Glenn Randall is a professor of microbiology and chair of the Committee on Microbiology at the University of Chicago, where for the past 15 years, he has overseen studies for emerging RNA viruses. This year, Dr. Randall was also appointed the Director of Emerging Infection Research at the Howard Taylor Ricketts Regional Biocontainment Laboratory, where he leads the lab's COVID-19 research. Last but certainly not least, Dr. Kelly Wrighton is a professor for soil and crop sciences and microbiome science at Colorado State University, where her research focuses on the chemical reactions catalyzed for microorganisms. Prior to joining Colorado State, Dr. Wrighton was an assistant professor of microbiology at The Ohio State University. So thank you to all of our witnesses for joining us today. As you should know, you will each have 5 minutes for your spoken testimony. Your written testimony has already been circulated and will be included in the record for the hearing. When you've completed your spoken testimony, we will begin with questions. Each member will have 5 minutes to question the panel. So we will begin with our witness testimony, and we'll start with Dr. Maxon. Dr. Maxon, please begin.
Chairwoman Johnson, Ranking Member Lucas, Chairwoman Fletcher, Ranking Member Weber, and members of the committee, thank you for including me in this important hearing. My testimony reflects my views only and not those of the Department of Energy. DOE's history of biological research is fascinating, from pioneering nuclear medicine and understanding the impacts of radiation on humans, to how biology drives energy solutions and creates new economic opportunities for the U.S. bioeconomy. Because of the foundation in biology built across the National Lab Complex, the Office of Science, Biological and Environmental Research Program, BER, is today one of the world's leading supporters of non-human bioresearch, that is, biology of microbes and plants. BER delivers transformative energy and environmental discoveries and solutions, and along with the broader Office of Science and DOE capabilities, can respond aggressively to national crises such as the current coronavirus pandemic. Berkeley Lab's founder, Ernest Lawrence, in 1931 invented the cyclotron, a particle accelerator that is the original ancestor of today's DOE light sources, the Large Hadron Collider, and particle accelerators around the world. Understanding the cyclotron's potential beyond physics, Lawrence asked his younger brother, John, an MD, to harness it for bioresearch, a move that has changed modern medicine forever and laid the foundation for DOE's biosciences capabilities. In 1937, John used radioisotopes from the cyclotron to successfully treat a bone marrow disorder, and later used beams of energized neutrons to treat leukemia, the first cancer treatment with beams from a particle accelerator. And with that, the field of nuclear medicine was born. Bioresearch wasn't limited to human health. At Lawrence's urging, Melvin Calvin and his colleagues used a radioisotope of carbon to trace how sunlight drives photosynthesis, winning a Nobel Prize in 1961. Because of BER's deep expertise in bioresearch and the department's role in large interdisciplinary initiatives, the nation turned to DOE and then later to NIH to sequence the human genome. DOE's part of the Human Genome Project was focused on a collaboration among 3 national labs to create the Joint Genome Institute, the JGI. JGI contributed 13% of the total Human Genome Project, and now managed by Berkeley Lab, is the largest facility in the world dedicated to genome sciences for energy and environmental solutions, with roughly 65 billion genes from microbes alone. And that's significant given the basis of almost every biomanufacturing process starts with genes and circuits of genes harnessed to make useful bioproducts, including fuels and therapeutics. Today, Berkeley Lab's Joint Bioenergy Institute, JBEI, a BER Bioenergy Research Center, has leveraged DOE's bio-expertise, facilities, and whole systems approach to lower the cost of bio-based isopentanol, which has an energy density close to gasoline. 10 years ago, 1 gallon of isopentanol produced in the lab cost about $300,000, and today it's closer to $3 a gallon. DOE is now able to respond to the coronavirus in similar ways to the Human Genome Project response and JBEI's systemic approach. That is, with diverse teams working together under the National Virtual Biotechnology Lab, established by Office of Science Director Chris Fall and directed by Deputy Director Harriet Kung. The NVBL has brought together all of the national labs to advance innovations in coronavirus testing, new targets for therapeutics, epidemiological and logistical support, and to address supply chain bottlenecks. The national labs are now leveraging DOE user and collaboration facilities to understand the ancient origins of coronaviruses, to identify possible COVID-19 treatments quickly, develop biomanufacturing processes for new therapeutics, and investigate new materials and reagents for viral detection. DOE's bio capabilities promise to give rise to future new tools to reproducibly study biological systems in controllable, fully instrumented lab ecosystem environments, something not possible today anywhere in the world. These new fabricated ecosystems are envisioned to help understand microbiomes and how they control soil carbon cycling, and could also be used to detect, identify, and mitigate new pathogens in soil systems. In summary, DOE's bioresearch enterprise has a significant history and an urgent, vital future for delivering scientific solutions to drive the U.S. bioeconomy. Thank you very much.
Thank you very much, Dr. Maxon. Dr. Mohnen, would you like to go next?
Certainly. Good afternoon, Chairwoman Fletcher, Ranking Member Lucas, and members of the subcommittee. It is my pleasure to respond to the 3 questions about the Biological and Environmental Research Program, BER, within the DOE Office of Science. First, why does the BER program have biological research and development activities and capabilities? That's a good question. One might ask, what does energy have to do with biology? And the short answer would be a lot. First, let's consider DOE's history. DOE was established in 1977 through a consolidation of more than 30 energy-related efforts in different government agencies, some of which were already doing bioscience. Thus, even at the time of its establishment, DOE was involved in bioscience. The origin of the biological research within the U.S. energy effort began during and after World War II with the Manhattan Project and the Post-War Atomic Energy Commission, and the development of an advisory committee to study the effects of radiation on humans. And this was later expanded to studies on the effects of radioactive fallout on the atmosphere, terrestrial, and marine environments and organisms. Thus, from the origin of DOE and the later form BER, they supported a combination of physical, chemical, and biological research. This was carried out by both DOE and academic scientists and facilities, and the goal was to meet the U.S. energy needs. Importantly, due to a mandate for DOE at the time, there was a formal division between the basic and the applied research. And the Office of Energy Research, later named the Office of Science, was given the task to oversee the basic research programs. And since BER is a part of the Office of Science, it supports and fosters critical basic science to meet current and future energy needs. In keeping with its historic roots, the current stated goal of the BER program is, and I quote, to support scientific research and facilities to achieve a predictive understanding of complex biological, earth, and environmental systems with the aim of advancing the nation's energy and infrastructure. It's relevant to today's hearing that the knowledge, tools, intellectual workforce, and facilities that BER has supported and developed over the last 30 years to meet the U.S. energy needs have provided cutting-edge scientific instrumentation, facilities, and expertise that can immediately be applied to national emergencies, such as the development of COVID-19 pandemic. As mentioned already, these capabilities include DNA and RNA sequencing, including the initial mapping of the human genome, and to date, having fully sequenced genomes of over 12,000 bacterial species, 3,000 viral, and 93 plant species. This is an enormous accomplishment. Importantly, BER has also supported the development of a systems biology approach and via the use of supercomputers and artificial intelligence to help understand and model complex organisms. Secondly, how are the BER-funded expertise and advanced research tools being leveraged to respond to the COVID-19 pandemic? The world-leading capabilities I've just mentioned, including the world's fastest computers, have enabled the BER-funded researchers to rapidly direct their attention to the national and global threat of COVID-19. The DOE capabilities being brought to bear include, I'll just mention two here, DOE structural biology resources, which have led, among others, to a new understanding of the three-dimensional structures and molecular actions of protein components of the SARS-CoV-2 virus, which helps us understand the disease. Another example is the work by DOE Oak Ridge National Lab systems biologist Dan Jacobson, who uses Oak Ridge supercomputers and systems biology to analyze the genome, the transcriptome, the RNA, the proteome, and evolutionary data from human lung samples of very ill people and also people control samples, as well as taking advantage of the data across the world. His team has recently published and is continuing to work based on these holistic analyses, a new proposed mechanism for COVID-19 infection, as well as multiple therapies using existing FDA drugs discovered through the systems biology approach. And finally, the future directions of the BER department. The importance of understanding and utilizing complex biological systems to meet our current and future energy needs is particularly evident when one considers that each year, more than 100 billion tons of carbon dioxide are fixed via photosynthetic organisms into biomass. And this biomass is essential. It's an essential large-scale renewable resource for energy, chemical, and biomaterials production. And when one considers that fossil fuels, which represent 80% of our current U.S. energy needs, are simply ancient biomass that was converted over time and pressure to petroleum, natural gas, and coal. Thus, the importance of understanding plants and microbes that produce and can transform this biomass into materials and energy cannot be overstated. And in conclusion, just as BER carried out biological research in the past to safely develop energy supplies, its future must take the next step in understanding and utilizing biology and biological organisms to ensure a continuing and strong U.S. energy portfolio. Indeed, the U.S. should lead the world in these efforts, the results of which will drive a new national and world economy. Thank you.
Thank you, Dr. Mohnen. Next, we'll hear from Dr. Randall.
Chairwoman Fletcher, Ranking Member Lucas, and members of the subcommittee, I Thank you for the opportunity to participate in today's discussion about biological research at the Department of Energy. As was mentioned, I am currently directing COVID-19 research at one of our country's 13 regional biocontainment laboratories. And so we'll focus my remarks as to how DOE is responding to COVID-19. So earlier this year, we established a SARS-CoV-2 Research Corps. And the idea behind this is that very few of these high biocontainment biosafety level 3 facilities exist. And there are many people with good ideas who don't have access to high containment. And so we provide collaborations where we provide both the facilities and the expertise to work directly with SARS-CoV-2. And this is primarily focused on evaluating treatments and vaccines, a little bit on the biology of the virus, but mostly translational. It's in this capacity that I've gained a real appreciation for the value of the COVID-19 research performed in the Department of Energy. In particular, I've enjoyed multiple productive COVID-19 related collaborations with scientists at the DOE's Argonne National Laboratory that I would be happy to discuss in further detail. But suffice it to say, we've identified dozens of therapeutics, both FDA approved and novel, that are active against the virus, at least in vitro. The DOE's Office of Science's Biological Environmental Research, or BER program, as has already been discussed, as a storied history integrating biologists, physicists, computer scientists, and engineers to address some of the important questions of today and tomorrow. Many of the extraordinary capabilities that BER has nurtured have been foundational to a specific response to COVID-19, which is the Virtual Biotechnology Library. This is a consortium of all 17 DOE national laboratories, each with core capabilities that are relevant to the threats posed by COVID-19. They leverage expertise and technology that synergistically interact with each other, academia and industry, to advance our fight against COVID-19. This effort capitalizes on long-held expertise in BER and unequaled strengths, particularly solving structures of proteins, what they look like, and how to target them with drugs or neutralizing antibodies, and supercomputing to stimulate billions of potential drug target interactions. This amplifies our current pharmaceutical capabilities by orders of magnitude. It is in these two areas that I have collaborated with DOE scientists and am most knowledgeable. I've worked together, as I said, to identify multiple drug candidates with DOE scientists. Other areas of NVBL emphasis include genome sequencing to track SARS-CoV-2 evolution and potential development of resistance to treatments, epidemiological and logistical support, protected databases that would host patient health data for research and analysis, manufacturing capabilities to address supply chain bottlenecks in areas such as PPE and ventilators, testing of clinical and non-clinical samples, and more recently, a project designed to address open questions about the mechanisms of SARS-CoV-2 transmission that will help inform approaches to interrupt chain infections and inform strategies that will guide our resumption to normal activities. The coordinated response of the NVBL to COVID-19 addresses critical needs in developing effective cures and vaccines that will help end the pandemic, and as applies to the future, will help provide a framework with how to be more responsive to the coming pandemics, because this won't be the last one. Thank you for your time.
Thank you very much, Dr. Randall. We'll now hear from Dr. Wrighton.
Chairwoman Fletcher, Ranking Member Lucas, and the rest of the committee, thank you for inviting me today. As you've heard about DOE's history of biological research, I'm here to tell you about how this history of pioneering biological research is active today and is perhaps best manifested by ongoing DOE investments in user facilities, including those of the Joint Genome Institute, or the JGI, and the Environmental Molecular Sciences Laboratory, or EMSL, amongst more than 20 other facilities. Although I'm not directly affiliated with these facilities, I represent the experience of a self-titled super user. Since my laboratory's inception in 2014, I've managed 9 different projects and awards with EMSL and JGI. From my narrative, I want you to take away a key message. These user facilities propel science in this country, and especially can benefit those like myself at the earliest stages of their independent research programs. Starting a research program at a university is much like starting your own small business. Essentially, your job is to take the university's investment in you, called startup funding, and use it to finance innovative science. The goal is the short-term investment by the university will enable one to obtain data and recognition to compete for external research dollars that fuel independent scientific endeavors. User facilities played a vital role in my early career by allowing me to maximize my startup investment. First, they allowed me to scale my scientific scope beyond what was possible in my new laboratory with a small, nascent workforce. Second, they provided me access to equipment beyond what was located in my building or even on my campus. Third, they networked me with experts who are at the cutting edge of their fields. My early collaborations with DOE user facilities led to scientific publications that developed me as a research leader in a few short years. More important, we generated data that facilitated my future fiscal independence, forging projects sponsored by US industry and the National Science Foundation. This symbiotic relationship between individual researchers and user facilities benefits the entire scientific community, because what it enables us to do is collect diverse data streams, and then this content is subsequently populated in databases that's shared with the community. In summary, DOE investments in user facilities are invaluable resources that amplify the innovation and extend the research dollars of our scientific enterprise. I know today when we talk about biology, we must address the dominant issue in public health, COVID-19. While DOE's direct contributions to COVID-19 research will be articulated and were very well articulated by other members of my panel, an area that I can speak to is this idea of translational investment. Essentially, how does investment in one scientific arena energize or cross-pollinate other parallel scientific discovery? You don't have to know biology or envision that detangling invisible microbes from wetland soils or shale rock is not the cleanest or easiest of work. On the environmental side, we have a long history of developing methods for isolating DNA and RNA from these complex matrices, technologies that is used by my colleagues doing SARS-CoV-2 surveillance research in wastewater and other systems. Moreover, even prior to the pandemic, DOE was leading investments in viral mechanistic ecology. From every habitat we explored, from deep below the Earth's surface, to our soils, to our rivers, and even our own guts, research from my group and others has recovered new viruses and demonstrated key roles for these viruses in modulating nutrient cycles. Currently fueled by DOE's support, teams I am part of are devising new software for rapidly detecting viral genomic signatures in environmental data, as well as defining the biochemistry enigmatic within these poorly understood viruses. In summary, DOE has developed the foundational expertise and technology in genomic sciences that can lead and be translated to epidemiological solutions for today and future public health challenges. Lastly, despite the advanced capabilities the user facilities shepherd, looking to the future, there are areas to reinforce our nation's capabilities. Currently, genomics information is being generated faster than the corresponding capabilities can keep up with, and more so than our computational infrastructure can mine. This means we have thousands to tens of thousands of genes that lack any known function. Or said more positively, this means there's a huge reservoir of biotechnological applications awaiting discovery. What 3 areas are needed to expedite the speed in which researchers can translate genomics information into actionable knowledge? We need a coordinated, organized computational infrastructure that enables computer-aided pattern recognition of this deluge of genomics and microbiome data. Second, we need research automation and scale that extend beyond the resources of any one lab and even those of our user facilities as they're designed today. And last, the heart of future discovery lies in creating this multidisciplinary, higher-risk collaborative space. In summary, this streamlined and cross-disciplinary scientific vision will allow us to embark on a new era of decoding biological information that heavily leverages DOE's genomic infrastructure. This trailblazing will result in new biotechnological innovations to environmental, engineering, and health-related challenges that will be faced by mine and subsequent generations. I thank you for your time.
Thank you, Dr. Wright. We will now proceed with our first round of questions, and I recognize myself for 5 minutes. This question that I have is really a broad question directed at all of the witnesses. So happy for you all to take this in any order you choose and to kind of share with us between each other a question about what has happened. Basically, in response to the pandemic, DOE launched the National Virtual Biotechnology Laboratory, NVBL, which strives to mobilize the resources of the department's 17 national labs to engage in COVID-19 research. I would like to hear from you all whether you think the NVBL should continue its work, even in the future when we are not actively responding to a global pandemic, and if so, why? And if you could touch on in your responses, maybe in what ways could the activities of the NVBL help accelerate drug development in general, pandemic, and how has the creation of the NVBL influenced operations or changed the partnerships between the national laboratories or with academia and the private sector? So I'd love to turn it back to the panel for your thoughts on that question. And maybe if we'll go back in order, we could start again with Dr. Maxon.
Thank you for that question. I'll tackle a couple of parts of it. The first being, do I think the NVBL should continue? I think there are a number of good reasons why the NVBL should continue, and a primary one being that it's likely that this will not be the only pandemic that we'll see, and it would be a missed opportunity not to have an NVBL poised and ready to tackle the next one. You also asked about how has the creation of the NVBL and the response to the coronavirus pandemic influenced partnerships? I can say, having been at a national lab for a number of years now, I've never seen more collaboration across the national labs working synergistically on a common problem with different pieces of it in ways that we are now doing as a consequence of the NVBL.
Terrific. Thank you, Dr. Maxon. Dr. Mohnen?
If I could be the last one to speak,
Sure.
I'm not directly influenced. I want to read up just a little bit.
Oh, absolutely. I will come back to you. Maybe I'll go to Dr. Wrighton.
Sure. I actually am not directly affiliated with NVBL as well, but I think when I was talking about this future of discovery and how we can catalyze and build around kind of a central unit and a theme, I think NVBL embodies that. And so it was a problem that we were faced with as a research community, and it took new multidisciplinary teams, and it brought together people that hadn't worked together before. And I really think that that's the heart and soul of this future kind of innovation is building these kinds of teams to address real-time problems. So I absolutely think that NVBL and others like it should continue in the future.
Okay, thank you, Dr. Wrighton. Dr. Randall.
Yeah, I'd like to first amplify what Dr. Maxon said, which is that there will be future pandemics. And I think there's a history of investing briefly out an emergency and then sort of forgetting. And so the anthrax attacks are an example of where we had biodefense apparatus that was heavily invested in for 10 years and then was no longer supported. And really, as we look at what works and doesn't work in our response to the current pandemic, that can really help form the framework of how we respond to the next one. From my personal interactions, the advantages are teamwork, collaboration, and really bringing forward a multi-pronged attack to address multiple disparate issues with this pandemic. In terms of immediate needs, I can talk about my own experience. We've talked a lot about the capabilities of the labs to solve protein structures. I benefited in that by the time we got SARS-CoV up and running here, our colleagues a few blocks away at Argonne had already solved structures of some of the most important proteins that are drug targets. Now we screen with them, give them the drugs, and within a week, they can show where the drug is binding to that protein, how to make the drug better, and how the drug works. It's spectacular. And the other aspect I would highlight is the supercomputing. So, the typical drug company has compound libraries of 1 to 3 million compounds that they'll physically screen. And so there's a consortium of supercomputing that have basically put together a virtual library of every chemical on Earth, 5 billion. So you're talking, or 3 orders of magnitude more, and can basically do machine learning and artificial intelligence to look at how these compounds bind to these drug targets. and then whittle that down to the top 1,000 or so, and then bring them over to my lab and we can test if they work. And quite a few do. And really, there hasn't been drug screening thought of this way before. And this is going to go way beyond COVID to any disease model, cancer, et cetera. So there's really a lot that can be leveraged not only for COVID, but for future advancement.
Thank you so much, Dr. Randall. And 5 minutes goes very fast, so I will thank all of you for your answers. Unfortunately, my time has expired, so I will now recognize Mr. Lucas for 5 minutes.
Thank you, Chairwoman. Dr. Wrighton, in your research, you leverage expertise from the Joint Genomic Institute and the computational capacities of the Environmental Molecular Sciences Laboratory. Can you talk about your experiences in working with both user facilities?
Sure. So I don't know how familiar everyone is, but when you want to access user facility resources, basically you write a grant. That grant gets reviewed by a board or review panel of other scientists. And you're basically, the DOE gives you a charge. So this year the theme is, and then they actually look at goodness of fit. So I've written grants, and I've basically been awarded many by JGI, the Joint Genome Institute, as well as at EMSL. I actually don't use their computational resources. I use more of their molecular resources. So they are innovators. I mean, they have access at both at Ohio State and Colorado State. We did not have the FTI CRMS capabilities, this mass spec capabilities that EMSL had. And so it was a great example of where I didn't have resources on my campus, nor did we have trained experts that could use that environmental data. But I could collaborate with EMSL, and I could get detailed information on the molecular structure of the soils that I was working in. And that they serve that role for many in the scientific community. And so it really is a way to enhance accessibility and to really expand your science beyond any boundaries that you may have on your campus or in your department. And the same goes for JGI. I mean, they're just the sequencing capabilities they have. I could maybe sequence 10 samples. With them, I've had hundreds to thousands of samples sequenced in a rapid turnaround time.
Speaking of accessing facilities, could you touch on for a moment about how the COVID-19 pandemic has affected your access to these facilities?
Yeah, it has not changed my access to these facilities per se. I mean, obviously, when there was lab shutdowns, these facilities were also shut down, but only in that sense. And I think that the facilities are really trying to make good faith to turn around and get samples processed as rapidly as they can. So I have not seen a change in my science in collaboration with those facilities due to COVID-19.
Dr. Maxon, can you give us your perspective from the laboratory side of how COVID has impacted access to user facilities at the Berkeley lab?
Yes, thank you for that question. Many of the facilities at the lab have remote capability. The supercomputing facility, the Joint Genome Institute, several of the data handling things obviously happen remotely. The Advanced Light Source has remote activities. However, there are things that need to get done by humans, and we are working very hard now to understand with safe what we call COVID controls, face coverings and distance working to protect the workers, and shift work, actually, which we never did before. We're trying to bring the full strength of the user facilities back online because we know the users depend on them. So we're doing our best to do that now, and we're not quite up to full speed, certainly not at the JGI yet, but we are definitely trying.
From your position now, looking forward, what do you expect in the terms of requests from researchers from this point on?
I think it will largely depend on what researchers can do with respect to collecting field samples. In the case of the JGI, we're looking at DNA that comes, nucleic acids that come from field samples. And if researchers are able to do the field work that generates the samples that then get sent to the user facilities, then I think it'll be a good response. I think we'll be fine. We'll be able to have a lot of users' needs met. If, however, the pandemic limits the ability of people to travel, to go to their field sites, I think there will be a reduced demand.
Absolutely. Dr. Wrighton, one more question. On top of being a frequent facility user, you also sit on the JGI advisory board. What would it mean for your research or others you hear from if BER's user facilities are not updated? Would the facility simply become obsolete?
Yes, I mean, absolutely. I think, especially, I think for JGI does a really nice job, and they have a call for early investigators. So you're not competing with people who have 20 years of experience. You're competing in a much smaller group, and they really train you in how to analyze the data and how to work with your data. So if those investments weren't made, I think that the biggest impact would actually be on the next generation of science and early career scientists, especially, because they do a really good job of kind of corralling scientists into in learning how to use the data and the technology, as well as giving access. So one of the neatest things about JGI is that they're always on the cutting edge. I mean, they set the methods, they're using the newest technology. And so I think that's what keeps them so competitive and makes them a place that really people want to come and bring their data and be part of, is because of the benefit of the cutting edge technology they offer.
Thanks, Doc. And Chair, my time has expired. I yield back.
Thank you, Mr. Lucas. I'll now recognize Chairwoman Johnson for 5 minutes.
Thank you very much. I'd like to start with Dr. Mohnen. As you noted in your testimony, much of the COVID-19 research BER is carried out and is built upon years of previous research. Can you speak to the importance of consistent and robust long-term investments in the BER program as a tool to fight future health and environmental crises?
Yes, absolutely. I've been working now in the capacity of the Bioenergy Research Centers with DOE and many BER-funded researchers for over 13 years. And it's very interesting to compare just briefly academic researchers versus DOE. DOE researchers are very much mission-driven. Academic, as was mentioned, you decide on the field or fields you're going to develop, and you do a very deep dive. And so what the DOE labs have been able to do is they take a mission-oriented, long-term approach on developing capabilities and improving them and do multiple things at once. They will attack critical questions that are mission important. And one of these, for example, with the Bioenergy Research Centers, has been to understand both on the microbial side and on the plant side the complex array of genes on the plant side that make the biomass and modify it. This has led to understandings that have allowed us to manipulate plants to get them to grow 6 times more biomass in the field. This has led to understandings of microbes that can produce chemicals. This has led to the development of systems biology capabilities and artificial intelligence to look at huge gene networks. And this research has both a fundamental and a potentially applied portion of it, and it develops a long-term commitment to build on the foundations that are established. So even though we've made, for example, great strides in understanding how to utilize biomass, deconstruct it, so to speak, convert it into various types of biofuels, we're still at a point where there is, again, as much that needs to be learned to make the kind of fuels that are needed for the future, to understand the involvement of the microbes in the field, to biomass growth, to be able to respond to climate change. I'm not sure, did I answer your question well? I could continue.
Yes. Are there other witnesses who'd like to comment on that? Okay, well, Dr. Maxon, DOE has a long history of supporting a variety of user facilities for use by researchers all over the world. In particular, DOE hosts several X-ray light sources that allow in-depth studies of materials at the atomic and molecular levels. Could you expand on how these light sources have been used to better understand COVID-19 as well as diagnostic and treatment options?
Thank you for the question. Yes, the X-ray light sources have been critically important. One of my colleagues on the panel mentioned that the X-ray light sources are being used to study in detail the specific proteins of the SARS-CoV-2 virus, how those proteins interact with the host. That's critically important. And so that's one simple example. Yesterday, I saw a fascinating presentation by a researcher using the advanced light source with soft X-ray tomography to look inside cells that are affected with SARS-CoV-2. What does it look like when they're not infected? What does it look like when they're infected? And how can we understand how the virus can hijack the internal machinery of the cell to make more and more viruses? And so I would say the light sources have very quickly responded to help not only identify the critical pieces of the viral proteins, but understand how the virus does what it does inside the host cells to make advances toward new therapeutics.
Thank you very much. I think my time is about to expire, so I yield back.
Thank you very much, Chairwoman Johnson. I will now recognize Mr. Biggs for 5 minutes. Mr. Biggs still with us?
I believe he's departed.
I believe he has, in which case I will recognize Mr. Cloud for 5 minutes.
Could you pass on me for the moment?
Yes, I can. I will now recognize Dr. Baird for 5 minutes.
Thank you. I really appreciate the opportunity to sit in on this session, and it's fantastic the work that these researchers are doing. Dr. Maxon, you just finished discussing how the proteins in the coronavirus, what they do in infected cells, and would you care to elaborate on that? I find that very interesting, how those proteins, DNA, RNA, the genome, and so on. I really would be interested in how the proteins in this coronavirus impact cells, lung tissue, for example.
Thank you for that question. So what I was able to learn yesterday in the study of the infected cells, it's still early days, so the experiments need to be worked out, and they're developing some results now. It looks like when the virus infects the cell, it then goes through a process of creating what's called a replication center. That replication center does what it sounds like it should do, and that is it begins to use the machinery of the host cell to replicate more and more and more parts of the pieces of the virus to create more viruses to infect, to then be released and infect other cells. It's really early, though, to be able to detect what the actual form of infection is that causes sickness. At least from these X-ray studies from the user facility, we're just a few ways off from understanding that. But understanding at the cellular level, the creation of a replication complex center, and the fact that there are cells that can fuse together to have two nuclei in the cell, that was found by these X-ray tomography studies. Very interesting and still very early days. We're not sure what it means yet, but getting closer to understanding it for sure. Thank you for the question.
Could I continue on with one? Can I continue on with one more question then? So how does these new proteins, how do they escape that original cell? Do you have a feel for that?
Yeah, so thank you. There's a process by which the cellular machinery is hijacked, if you will, not only to make more virus, but to extrude the virus out of a cell. This, for decades, we've understood how cells are infected with other types of viruses to then release the virus particles into other cells.
Very good. Dr. Wrighton, you mentioned your work in getting a lot of sequencing done in a very short period of time. But my question to you is, what happens to the coronavirus as it comes in contact with soil?
You know, I think that we're still in very early days in terms of surveillance of the coronavirus and other viruses like the coronavirus and their distribution across different ecosystems, soils, rivers, wastewater streams. I think a very active and exciting research that's led by some colleagues at JGI that I was actually just speaking to this morning about this was we're basically trying to figure out ways that we can survey the diversity of these types of viruses so we get a sense for the reservoirs of these viruses. Also, we're trying to develop new tools so we can look at the variation within these viruses so that we can maybe start seeing these different populations and these changes, and we can maybe better track these viruses using their genome tags over time and space beyond just the human host, but have a better, broader environmental context. So I think that's a place where JGI will really play an important role moving forward.
So I got about 1 minute left, and I would ask you and Dr. Maxon both. So the BER, in your opinion, plays an important role in finding the answers you just both discussed.
Yes.
I see you nodding your head.
Yeah, without a doubt. And I just think too, it's this parallel investment. I mean, I think anytime you get discovery on one end, it transcends and it fuels another side and back and forth. And I think that's what we really need to be armed and ready for this pandemic and the next pandemic.
Well, I think that kind of cooperation and collaboration is absolutely essential. And I think this basic research is really critical, especially in times like this pandemic. So I see my time is about up. And so, Madam Chair, I yield back the balance of my time. Thank you.
Thank you, Dr. Baird. I'll now recognize Ms. Horn for 5 minutes.
Thank you very much, Chairwoman, and thank you to all of our witnesses for this this insightful and incredibly helpful hearing today. My first question is for Dr. Maxon and Dr. Randall, and specifically around DOE's laboratories and their involvement in past pandemic responses, such as HIV, Ebola, and influenza. And I'm wondering how the existing research has been adapted or reoriented for COVID-19 research purposes right now.
Yeah, I can say, in particular, the two past coronavirus pandemics, SARS-CoV-1 and not as big, but Middle Eastern coronavirus. We're talking about how these proteins look, their structures, and they're similar and they have similar biochemical properties. So knowing how we could make and purify them and what they look like really expedited how fast we could learn the structures of the current coronavirus, SARS-CoV-2. So that's certainly one example of where past research really had us prepared and ready to move very quickly with the current pandemic.
Very much. Dr. Maxon?
Thank you. I will offer one example. I know that from the time of the Ebola virus pandemic, the advanced light source researchers, again, used the X-rays to understand the viral structure and how the proteins of the virus do what they do. So I do know that at least in the case of the Ebola, the Department of Energy was in fact involved.
Following up on that a little bit more, Dr. Maxon, how has the COVID-19 pandemic differed from outbreaks of other infectious diseases in terms of impact on DOE's research efforts or the way that DOE has approached disease-specific research?
Impacts. There's a couple of ways. I think first, I would be remiss if I didn't say that a major impact of the COVID-19 pandemic is on the costs of doing research. That has been a significant challenge for us to deal with. So that's one. I think in terms of impacts of disease research specifically around this pandemic, as I said, bringing together the labs to pull all parts of what we have as core capabilities toward this problem. We had people working with the parts of the lab that do biomanufacturing process development. Never before working on a treatment for an antiviral, but definitely doing that now and working with companies to do it.
Very much. And I want to turn slightly different focus for just a moment. And Dr. Maxon, continuing on with you. In your testimony, you mentioned that DOE research has drastically reduced the predicted cost of new biofuels. And as we are looking at not only addressing the next pandemic and DOE's role in research, we also have to take into account so many other factors, environment and related factors. And biofuels are going to be a critical component of, I think, next generation energy. So I'm curious about what advances or how these advances are transferred to industry, and what additional resources may be needed by DOE to help enable the commercial adaptation and adoption of biofuels.
Thank you. Biofuels. So the way that these advances are translated to industry include from the Bioenergy Research Centers proactive engagements with industry to make clear that there are new technologies available in the biofuel space for licensing, frankly. And I think what's required now, there's still a gap, as I mentioned. The costs have come down, but the gap in being able to make these commodity products, these biofuels at scale, is missing. And that piece, being able to take a small scale laboratory proof of concept and make it commercially scaled, that's the gap that I think is seriously missing, and we could use some help.
Very little time left, but thank you for that. And I think that filling that gap is critically important. So thank you. And Madam Chair, I yield back.
Thank you, Ms. Horn. I'll now recognize Mr. Cloud for 5 minutes.
Thank you, and thank you all for being here today. We certainly appreciate the work you do to keep us on the forefront of science, especially when we consider the competitive global environment that we're in, how important it is for the United States to stay on the cutting edge of these technologies and these advancements in science. Really appreciate it. Kind of continuing on with the questioning Ms. Horn had, just talking about some of the lessons learned from previous pandemics and such. Not only are we learning a lot about the science when it comes to COVID, but it seems to me we're doing things a lot differently. Not only coming up with the new discoveries, but also new best practices. Could you compare maybe some of the lessons we're learning from a operational standpoint, from a best practice standpoint, compared to how we approached the work of research compared to previous pandemics?
Years with people that use systems biology and computational modeling on plant systems, where we don't have as much information as we do on human systems, because on plants there are many, many species. Humans, we've got a lot more money and concentrated on humans and model mice, etc. So the data that you have for human systems is incredible. Whereas a systems biology approach is always limited by the data set. When you get to humans, what I've seen now with what Dan Jacobson and others can do with these supercomputers that DOE funds, we've got the second fastest in the world, and the amount of data out there, both published and in-house, from the DNA sequencing, RNA sequencing, et cetera, they are now at a point I actually didn't believe a couple years ago we would be at. They can make predictions by running supercomputers and integrating all the data from metabolomics, from proteomics, genomics, evolution, and they can come up with hypotheses that have a very strong potential of being correct. That can direct our thinking. We've gone over, I think, an edge to where now you're not going to get definitive answers from this, but you're going to get answers that are highly probable and then can inform the people that go in the lab and do the experiments. I think this is a turning point that only has become possible with these supercomputing abilities and the ability to do the systems biology. And finally, the fact that you've got this national lab set up where you interact with a bunch of specialists, whether they be academic or in the labs, to inform the information as the results are interpreted. I'll stop.
Well, thank you.
Yeah. I was just going to follow that up and say I agree completely, and also the speed of sequencing and so forth has ramped up so fast that within discovery of the virus, we had sequence within a week, and all the companies that are rushing out their vaccines then knew how to synthesize spike to get it in their vaccine platforms, to where we're getting these vaccine candidates years before we traditionally have. And so and these platforms were all developed, for things that were not SARS-CoV-2. The only thing SARS-CoV-2 related in them is the spike protein. And so really there's a lot of platforms and best practices in place that I know the pandemic seems long, but the response historically is very fast.
Yeah. Wow. Well, thank you. Dr. Maxon, I was wondering, from your experience in Berkeley National Lab, we can appreciate the research going on, but then we also know that our research has been under attack in a sense from other nation states, specifically China. Can you speak to what the DOE has been doing to ensure that our research continues to be safe and secure?
I can speak from the perspective of an employee at Lawrence Berkeley National Lab.
Right.
We definitely take very seriously the export controls. We follow those controls very clearly to make sure that that our research stays our research. We are looking very carefully at our foreign visitors processes to make sure that we're making sure that we know who's coming onto the lab and we know what they're there to do. And so we're taking the precautions to make sure that our research stays our research. We've actually, in the last couple of years, updated some of our badging systems so that we can keep track of who came in and when. And so I think we are, at least at our national lab, and I'm sure others, are very, very concerned about keeping things and all of the data that we have in our labs secure from attack.
Well, especially with teleworking, I guess that's one of the concerns I had. Can you speak to that at all?
I understand. Thank you. Teleworking does present some new concerns, especially many of the computing people don't have home systems that can handle the big scales of data that they need to use. But as it relates to the security of the data, I understand that the IT infrastructures at the labs are working hard to make sure that we have all the right up-to-date tools. We talked about updating facilities earlier. Updating the cyber aspects of the labs are important too. Just for that reason.
Thank you very much. Appreciate it. My time has expired. Thank you for being here today, all of you.
Thank you, Mr. Cloud. I'll now recognize Mr. McNerney for 5 minutes.
Well, I thank the chairwoman and ranking member, and I thank the panelists. I have to say it is exciting hearing about what's going on in the labs. And my first question is for Dr. Maxon. It'll be a softball. And it's good to see you here, Dr. Maxon. You noted that societal challenges such as the need to store carbon at massive scale and reduce crops and develop crops for changing climate demand quick action and quick response. How important is it for the United States to maintain its lead in these areas?
Thank you for the question, Doctor. It's critically important that the nation maintain our leadership. We have the capability to produce 1 billion tons of sustainable biomass in the United States. It's a strategic natural reserve of sorts. And to be able to convert that biomass into the bioeconomy's products, including transportation fuels and chemicals, and reduce greenhouse gas emissions, it's critically important that we maintain that lead. We're the only country that has a lead like that.
Is the current federal investment adequate to ensure that we do maintain the lead?
Well, that's a challenging question to answer. I would offer from my own perspective that more resources would be very helpful in allowing us to understand how to take diverse feedstocks, such as agricultural waste and forest waste. In California, as you know, we have a lot of forests that are overgrown. If we could turn that forest waste, in that woody biomass, into biomanufactured products, fuels and chemicals used regionally, for example, like microbreweries, I think that would be a very good investment to make, more about how to change the feedstock capabilities of the United States.
Well, thank you. The pandemic has been with us since February or March, and we hear about the capabilities of the lab complex to address national crises, such as the pandemic. Can anyone on the panel point to specific achievements in this effort that is now helping the nation fight the pandemic?
Well, Mr. McNerney, as I mentioned more so in the written statement, I was actually very surprised when I read Dan Jacobson's work, because I work on the plant and micro side, so I had to catch up. That systems biology approach identified 11 FDA-approved medications that should be able to, if the analyses are correct, improve some of the effects of the COVID infection. And I assume those are being looked at immediately in small clinical trials. There are other people on the panel who are more experts in the COVID themselves. But even that, and his work's made quite a splash. It's been covered in Forbes and in many medical journals. And that's just one example. Then the other example was the work, and I've forgotten the researcher's name now. It comes out of the laboratory where they used the modeling capabilities and the 3D protein structure prediction. They, these researchers determined the first structure of one or more of the COVID proteins at a normal room temperature, at a temperature it might exist, and at the temperature of the body, actually. And that gave information on slightly different structures that could be important in understanding how medications or cell proteins or metabolites interact with them. So the results are completely new, up-to-date, and informative, but I yield now to people with more expertise with COVID-19.
Well, I'm going to move on to my next question, though. I'm wondering how the lab consortium or collaboration have been working with the pharmaceutical companies to develop vaccines. You just mentioned, Dr. Mohnen, about therapeutics. Is anyone able to give an example of collaboration between the BER and private companies, and what are the ownership issues involved? No one's going to bite on that one. Okay, well, my last question. On Wednesday, 78 Stanford researchers and physicians issued a letter about the falsehood and misrepresentations of science that have been spread by Dr. Scott Atlas, who was appointed to the White House Coronavirus Task Force last month. This is only one example of the administration's attack on science. What impact do these disregards for science have on our ability to fight COVID? Whoever wants to step up.
I'm not going to speak specifically to that, but what I will say is that public trust in science is critical to get people to take the vaccine when we get it. And that is a concern.
All right, thank you. I yield back, Madam Chair.
Thank you, Mr. McNerney. I'll now recognize Mr. Foster for 5 minutes.
Thank you, Madam Chairman, and to our witnesses. So I have one very specific question and then one much more general one. The specific one is that there is this theory of very severe cases of COVID that goes by the name of the Brady-Kinnon-Storm hypothesis. And this apparently was discovered or verified using the Oak Ridge supercomputers, where they analyzed the fluid in coming from people's lungs who were very sick with COVID and looking for genes that were massively overexpressed, and saw that the genes involved in, what do they call it, the RAS system, which is local inflammatory response and blood pressure regulation. And so this apparently, this hypothesis, which was verified on the Oak Ridge supercomputers, explains everything from COVID toe to the fact that the virus gets in through the blood-brain barrier, to the fact that vitamin D is a very promising therapeutic and prophylactic. I was just wondering, is that on any of your radar screens? Is that a real result coming from the DOE supercomputers? Anyone familiar with that?
Yeah, it absolutely is a real result. It comes from Dan Jacobson's work, and he's got multiple papers out. I've been reading a couple of them. He is the top-notch, world-class systems biologist. I spoke to him personally for this panel. I know him. He works in the Bioenergy Center. He works because he's so good at what he does. And he's done two, it's either the largest or the fastest computational predictions using computers anywhere in the world. He really is outstanding. I talked to him about this because I couldn't understand how he did it. And he told me, because he had to keep up his other work, which was on microbes and plants, as this hit, and he knew with the systems biology approach and the computers, because this is what he does. He takes multiple pieces of data, uses the computers, looks for connections, then reads the literature deeply. He was working 21 hours a day for weeks and weeks on end. And he brought in researchers from multiple medical people from multiple institutions. Now, what we have to say is when you do this kind of systems biology approach, and the data look very compelling, and he will say in his paper, now it has to be tested. These have to be tested in clinical trials, but the data are incredibly robust. And I believe it's all being followed up, and I know there are many more papers. So this is
his paper identified a number of therapeutic targets, and I presume those are being followed up in clinical trials, though I'm not familiar with that.
I'm not sure. I believe so too.
Yeah. So that, all right, my more general question is, one of the trends that people mention in biology is this business of what's sometimes called cloud-based biology. And that's where you have large farms of robots that will do biological experiments. And so this is something where potentially, a scientist at a university can sit down in their computer, define the experiment. We want to, get this cell line and modify it genetically this way, expose it to this, wait 18 days, and then, section it up and send me the photos. And so without actually ever touching or owning or taking possession of the biological samples, could perform experiments. And it strikes me this is something where there may be a role for national labs to actually engage in gigantic purchases of initial systems, the same way we engage in new generations of supercomputers, the new generations of experiments, and then open it up, in a way very similar to supercomputers, where different university groups can sort of bid for time on these things. And I was wondering, is that a sort of model that makes sense to seed investments that can be immediately used by universities? And this seems to relate to Dr. Wrighton's comments earlier.
Yeah, I'm so excited, Congressman Foster, to hear you say this, because this is exactly up my alley. I mean, I think that what we're finding at the user facilities is they're very successful, but many of the key resources are becoming fairly inundated. So the turnaround time or the life cycle of actually processing samples is somewhat delayed just because of the demand. And so we really have to rethink scalability in terms of maybe not one building, but maybe and there are some companies that you may be familiar with, Emerald Cloud Labs and others, where you can basically, they have these robotic facilities and you can log in and kind of high throughput with more reproducibility and greater efficiency. And so that will really allow us to run little pilot studies, look at the data in real time, and then do bigger experiments. And so it creates a much more dynamic and efficient working environment than having to collect 400 samples and send them all in, because that's the allocated time you get. So I think the future, if we're really going to talk about how we can innovate and do more with what we have, or even just extend our resources in new ways, I think that's a very exciting future.
Yeah, and it would allow smaller university-based researchers to compete at the very top level, as well as dealing with the reproducibility crisis, some fraction of it, at least in biology, that you'd publish the specifications and could reproduce that at any robotic facility anywhere. I see my time is up and yield back.
Thank you, Mr. Foster. I will now recognize Mr. Kasten for 5 minutes.
Thank you, Chairwoman Fletcher, and thanks to our panel. This is going to surprise all of my colleagues, but my questions are about climate change, not COVID. I have, and number one, so not lead to any conclusions. We have got to get to zero net carbon emissions, and we got to get there yesterday. I am somewhat sanguine about our path to get there in terms of our energy use, because I can identify technologies to make electricity, transportation, fuel, heat, the things we need for energy. I have real concerns about what we are going to do in those places where we use fossil fuels as a chemical input, typically to reduce organic compounds. How do we make fertilizer? How do we make steel? How do we make silicon? How do we make magnesium? And biology has a way to do that, right? The photosynthesis is a way to reduce compounds with sunlight input. Some of the weird archaebacteria that live on volcanic vents do that. And it strikes me that there are interesting research projects that are scattered. I introduced a bill that's passed through this committee, HR 4320, the Clean Industrial Technology Act, which has a purpose to bring all of the DOE research around how do we decarbonize those hard to decarbonize industries and bring it to one place. And I guess I want to start with you, Dr. Maxon. Can you give us any oversight of what, if any, programs you are aware of that DOE is doing in that vein around how do we biological solutions to reduce inorganic materials? And what, if anything, can we do to help accelerate that research?
Thank you for the question. Biomanufacturing, yes. In fact, the Department of Energy in January hosted an Innovation X Lab Biomanufacturing Summit directed on this very topic and invited hundreds of companies to come in and see the technologies, the assets, and the programs that the Department of Energy has at its national labs to do just this, to reduce the energy intensity of manufacturing and to use petroleum, to reduce petroleum feedstocks. I mentioned a little while ago the billion-ton bioeconomy, the billion tons of sustainable biomass. We're making big progress in being able to convert that biomass into useful things, but can't do it cheaply yet. There are a number of other programs. The Agile Biofoundry is a Department of Energy program that is looking to speed for industry and for academic partners the ability to design biological systems to do just this, to harness them to do biomanufacturing. So the Agile Biofoundry is a new one. The bioenergy research centers are focused on lignocellulosic, conversion of lignocellulosic products, woody biomass, for example, into fuels and bioproducts. So there are a number of them that exist, but I would say that given the crisis, we could use a lot more help in this regard, more of these and more pilot fermentation facilities, steel, if you will, fermentation to get us to that next scalable leap.
So if I could, and maybe others, I'm not sure I've asked my question very well. The lignocellulosic materials, making building materials, making products, I get that. I don't even understand the thermodynamics well enough, but if we're going to take, nitrogen and make it into ammonia, I know how to do that with natural gas. If we're going to take quartz, silicon dioxide, and make it into silicon, I know how to do that with coal, or to reduce iron oxides into steel. Are there biological pathways that we could imagine to use biological systems' ability to reduce those compounds? And is there potential to scale that up, or is there some reason why those are just, is there something about, the way that life is put together or the thermodynamics that makes that impossible?
I think, Congressman Caston, there's two parts to this answer. One is the biological discovery angle, and I think, just 2 weeks ago, we came out with a paper looking at how you can produce ethylene with microbes using enzymes that we never knew about until just this year. And so, and do it independent of oxygen, so do it independent of combustion. So I think that there is this discovery aspect and this basic science aspect, but the scalability part, I think, is the yet to be seen part for me in terms of harnessing these new biological pathways and overcoming the thermodynamics within an organism, and then thinking about how we actually can develop these precursors with, a more neutral carbon economy. But I think that we're at the point now where we're able to now mine and scratch the surface into the biology. And the next phase of our discovery is going to be scaling and do these technologies, at least in these really new spaces, not some of those like plant deconstruction, and they're 10 or 15 years ahead of some of the more new discoveries we're finding about microbial pathways for these compounds.
Okay, well, I'm out of time and yield back, but we'll maybe follow up with you offline, because I'd like to get some overview of what these programs are and what we can do to accelerate, because I do think we're out of time and we need to.
Okay.
By 5 minutes, we're out of, our species is running out of time. But we'll continue that offline. Thank you.
You are correct, Mr. Caston. Your 5 minutes has expired, and we will now recognize Mr. Byer for 5 minutes.
Thank you, Madam Chairman, very much. And thank you, panelists, for amazing amount of information. I'd like to start with Dr. Mohnen. And thanks for your comments about Dr. Jacobson and the work done at Oak Ridge. I want to thank Bill Foster for sending that article around, which I thought was just remarkable. The notion of crunching data on 40,000 genes, 70,000 genetic samples, 2.5 billion genetic combinations. And the articles themselves really lead to some interesting thoughts about treatment. If this Brady-Kinnon storm thing is real, we could be in a very different place in a couple of weeks from now. But Dr. Mohnen, you're also the historian. So I want to thank you for letting us know that the Human Genome Project started through BER. And I wonder, because I've always thought, Francis Collins, Craig Venter, George Church, how now does JGI interact with NIH and with the other folks doing human genomes?
Well, thank you for the question. I think it's a very interesting one, but I think that there are other panelists who probably have better information on that interaction. So I think I will pass that off to someone who knows more about that particularly.
Dr. Wrighton?
So the question was, sorry, could you repeat your question?
Well, so you have the Joint Genome Institute, but then you also have NIH and Francis Collins, Nebula Genomics, all the work being done at Harvard, MIT. How do all those work together?
Yeah, and so I think, so to my knowledge, and others may be able to have a broader perspective, is that the Joint Genome Institute generally focuses on the non-human aspects of the biology. That does not mean that they're decoupled from NIH, and I want to stress that, because there are themes that you can do in one system, like understand enzymes and pathways and make discoveries, and then you can take those to the NIH, and I've done this in my own career, and apply for funding in the NIH and study those same processes in the human gut. So it's not that they're, but the research focus of the JGI is typically on non-human, research, but it doesn't mean the discoveries that are made at JGI by JGI researchers or their collaborators do not then go and advance it through the NIH angle. So they're just kind of different themes, but you can take ideas from one area and bring it to the other and vice versa. Does that make sense?
It does. And you set up my next question, which I think is for Dr. Maxon, because you're the federal lab person. Just reading your testimony and listening to it, I came up with the CABBI, the CBI, the GLBRC, the JREI, the JGI, the PRC, the NMDC, the APBDU, the EMSL, the KBase, the ABF, and others. Is this the idea that we have micro-focused so many different places, or is this empire building, or why do we have such an incredible proliferation of separate institutes within the Department of Energy, just on biology?
Thank you for the question. The biological challenges are enormous and they are complex. And each one of those 4 bioenergy research centers that you mentioned is working on a different way to attack the similar problem. And so it's a way to have non-redundant maximum shots on goal to achieve the solution rather than putting all your money in one basket. Going back to your question about NIH for a second, I'd like to weigh in on that as well for just a minute. And that is to say that the National Microbiome Data Collaborative is a brand new DOE program that is intended to take all microbiome data, as Dr. Wrighton was just talking about, and make that data fair, findable, accessible, interoperable, and reusable, meaning it doesn't matter whether the microbiome data came from NIH researcher or a DOE researcher or a USDA researcher. All those data, based on the vision, should be able to be findable, interoperable, and used together to develop all new theories and hypotheses and experimentation programs. So that new thing is helping to bridge the gap.
That's terrific. And one last question, Dr. Maxon. We have at least 3 prominent women scientists on our panel today. Does this mean that women are finally assuming their rightful place in science?
Wow. I'll say yes.
Okay, great. Madam Chair, I yield back.
Thank you.
Madam Chairwoman, I yield back.
Thank you, Mr. Beyer. Great last question. And it is wonderful to see the expertise assembled on this panel, the incredible women here, but really the efforts of everybody at our national labs and the work that you have done in response to the coronavirus pandemic and more broadly. So it's really wonderful for us to hear from you this afternoon. So I thank you all very much for your participation, for your insights, and for your work for our country right now. We need you, and we are so lucky to have you. So thank you so much for your testimony here today. Before I bring the hearing to a close, I just want to let my colleagues know that the record will remain open for 2 weeks for additional statements from members and for any additional questions that the committee may ask of the witnesses. With that, the witnesses are excused, and I'm going to use my gavel here so you can all hear. The witnesses are excused and the hearing is adjourned.